All right, we'll keep moving along. Our next talk is about breaking the diffraction limit with SciPy and Python. I'd like to welcome David Baddeley.
Okay, thank you very much. And I guess I should give some background about myself. I'm a physicist by training and working in biology, so I feel a little bit out of place here. But I'm going to spend about half the time describing what the problem that I'm trying to address is, because it's very different to the other ones I've seen, and then the rest of the half talking a little bit about how I've used Python to address that. And I recently moved to Yale, but most of the work I'm showing was done in Auckland University in New Zealand. So anyone with a physics background probably knows that how tightly you can focus light is limited by the wavelength, and that puts a practical limit on the resolution of any imaging system. So you can only, in a light microscope, you can get a resolution of about 250 nanometers in XY or 600 nanometers in depth. Now, if you want to look at a cell, that's sort of smack bang in the middle of where a lot of the interesting processes are going on. And so if you actually want to know how the machinery inside a cell is working to make your biology work, a lot of that is single proteins about 30 nanometers, and where you can sort of see something like this, where we sort of see roughly what the constituents of a cell, or the macroscopic constituents inside a cell, but we don't know how they're put together, or we can't see how they work. And the conventional way to get around this has been to go to shorter wavelengths and go to electron microscopes. And they give you better resolution, but they have a number of problems. They're sort of hard to use, and they only work on dead samples. But the most important problem, I think, is that the data can be very hard to evaluate because you don't have specific contrast for different markers. So people like to do light microscopy, because light microscopy, you can sort of pick two proteins that you want to look at, put a colour on them, and then you just see those colours, and you've got lots of contrast, and you can do it in live cells. And recently, in 2000, well, there's been a few methods, but the method that I'm going to talk about, somebody invented in 2006, and you can get a tenfold improvement. You can get down to sort of 20, 30 nanometers using light. And a tenfold improvement is the difference between being legally blind and having 20/20 vision. So a tenfold resolution improvement's quite a big deal. Now, I wish I could claim to have invented this, but I didn't. But the way people came up with for getting around this resolution limit is that normally in microscopy you look at, say, all the fluorescent molecules you have attached to your sample at one time. If, however, you can switch most of these molecules off, you can get an image where you see just a handful of molecules. And this is very like sort of images of stars in astronomy. You have point objects, and you pretty much know that each one of these point objects comes from one single molecule. And because you know it comes from one single molecule, you can go and work out where the position of this molecule is. And it turns out you can work out a position with better accuracy than your resolution limit. You see a blurry blob, but you find the centre of it, and that just reduces to a fitting problem. And if you can look at one subset, and then you can switch different sets of molecules on and off, you then sort of get a video where you're looking at the positions of multiple different molecules over time. And that means that you can accumulate a large number of molecular positions rather than a single image. And this is sort of what it looks like. This is some real experimental data where we have the positions where we've observed these single fluorescent molecules. And this is what it would look like in a normal imaging system. But if we then take these positions and do some form of kernel density estimation, we can get what is effectively an image with much, much better resolution. And what's made this possible, and what a lot of the challenges are in doing this, I missed out a slide here, and by doing this we can get new biological insight. And so this is an example of one protein cluster that we've looked at, and we found a number of things that hadn't been previously observed. And there's a lot of people doing this now, and it's one of the big new sort of fields that's taking off in biology, is this super resolution light microscopy. But because you have essentially a computational problem, well, the demands of doing this are mostly computational. And so you've got a lot of tasks. You've got to control your microscope. You've got to deal with the stream of video data, which is coming at moderate to relatively fast data rates, depending on your acquisition. Anything from sort of 10 megabytes a second up to several hundred megabytes a second. And you then have to analyze this data to find your positions of single molecules. You have to, and we like to do this in real time, because ultimately that's going to be a backlog. It's not much use generating a lot of data if you can never analyze it. And so biologists were using a microscope all day, and so you don't want to be sort of backlogging this data. And then you have to do some interpretation, visualization, and also understanding how things work. We tend to do a bit of simulation and some database stuff to keep track of it. And I've got software which does pretty much all of these aspects, and it uses Python and scientific Python tools in many different ways throughout the software stack. And so I've used a lot of the different bits of SciPy and NumPy. And I've been learning as I have gone along. And so some of the approaches I've taken have been pretty poor. Some of them have been better, hopefully, recently. And so if we look at the first problem, to try and do this real-time analysis, we want to parallelize the data. And for each sort of single reconstruction, you're looking at sort of 20 to 50,000 single images. And so this is a moderate scale parallelization problem. It's not a huge scale parallelization problem, but it helps to sort of distribute it either onto a really beefy workstation computer over several cores, or over a handful of machines, say 2 or 3 machines. And that can be accomplished quite nicely with just a sort of task queue architecture, where you sort of have worker processes grabbing queues. And I implemented this using Pyro, which makes it very easy to do, but has some sort of overhead. It's not necessarily what I'd choose to do it again, but it has some nice features, and it's easy to make a relatively simple queue-based task distribution setup. And so we've got the architecture here to do this, to control a microscope, push the images as they're acquired up to a server, and then distribute them out to workers. And this could be useful, the architecture could be used for anything where you have the sort of a stream of images coming in which need to be analyzed in real time. And so what I'm going to jump to now is an actual live demonstration, fingers crossed, of some of the features. And once you have this data, which is essentially a list of coordinates where you found single molecules, you actually need to do something more with it before the biologists can understand it. And so what you've got is a viewer here which allows you to explore this sort of 2 or 3D point data set an OpenGL, so it is relatively fast for hundreds of thousands of points, and you can sort of choose different attributes to color by. So this is actually a multi-color data set where you have 2 color channels, so you can choose to color by color or by the 3D Z position, or a number of other things. And there's a few neat widgets which allow you to adjust color maps quite easily as well. And you can add filters if you want to, say, restrict it to just one of the colors. We'll make that. And then you can get one of these nice things and say, and that will just restrict to one of the colors. And so it's a useful tool for manipulating data. And you also want to generate a biologist-friendly image. You want to go back to something which resembles a conventional image. And so this is basically kernel density estimation. And there's a number of different ways of doing that. And the one that we quite like is to do a triangularization of the data and then to jitter these points slightly and generate multiple samples in the triangularization. And then that gives you something which gives you a kernel density estimation while preserving edges and also taking account of the fact that you have vastly varying densities of your points across the image. And so you've got a bit of a view here, and you generate an image from your data which looks very much like a conventional microscopy image. You can also do this in 3D and with similar results. Now, this is all a little bit domain specific. I suspect there could be some other uses in sort of point cloud data, but hard to tell. What is potentially more useful for people who are not doing super resolution microscopy is got some general purpose 3D image data set evaluation code in Python. This is basically an ND array viewer which allows you to quickly scroll through 3D data sets, allows you to do things like X, Y, and XZ slices in it, change color tables, and zoom, and this works quite well for large data sets. And it was designed initially to deal with the raw data sets of the localization problem, which can be several gigabytes. And so it uses a lazy loading scheme. So it only loads the frame that you're actually looking at the time. And so it's hopefully useful. And it's got a lot of ImageJ-like functions in it, like doing basic filtering and thresholding and co-localization and all those other things that biologists tend to use ImageJ for. It doesn't do nearly all of it, but it allows you to sort of stay in Python for a little bit longer. And there's also some neat stuff in here like deconvolution, which works in a relatively intuitive fashion. And we'll, fingers crossed, we'll deconvolve your 3D data stack for you while you watch it. And this is a relatively slow computer, but it's a lot faster. Another one, this is, because it's designed to be sort of usable by biologists. There's a lot of, the API is still relatively immature, but there's a lot of goodness in there. And you can, it's got an easy interface into Mayovi, if that. So you can do 3D isosurface renderings and right, let's just okay, that's so yeah, so that's some of the functionality which I think should be easily usable. And then another aspect which I'd briefly like to mention, which has been very helpful to me, is a MATLAB style colored profiler. So you run your code and it prints out or produces an HTML file which shows which lines took all the time. And this has saved a lot of time with the optimization and just sort of getting a very visual representation of where your code is spending all its time. And this is not the first line optimizer in Python by a long way. Richard Kern's got one, but I like the graphical interface to it, and it's not the most efficient either, but it's yeah. So in summary, in this package, which is up on Google Code, there is microscope hardware control, supports a number of cameras, and a number of different sort of piezos and translation stages and kind of things you have on a fluorescence microscope. And it streams data to disk in HDF5 format, and there are also some neat code for simulating microscopes. And we have a sort of package for the super resolution microscopy for localizing point-like objects in 2D and 3D when they overlap, and also doing some neat color stuff. And for the visualization of these and exploration of this data, and some general sort of image processing type functionality like the deconvolution and the image viewer, and there's a few other things like sort of registration and segmentation in there as well. So, yeah, I'd like to thank you for your attention and acknowledge the people back in Auckland who provided me with data and samples to look at on the microscope, and lots of sort of help finding bugs and feature requests and stuff, and funding and collaborators, and yeah, thanks. Yep.
Do you implement any prior knowledge into the deconvolution?
At the moment, that's just got a positivity constraint.
Can you repeat the question?
Okay. So the question was whether I introduce any prior knowledge to deconvolution.
You can imagine calibrating a deconvolution filter based on the microscope in question.
Oh, absolutely. The deconvolution. Okay. So the deconvolution uses a measured microscope point-through function. And so you measure the point-through function of the microscope, and so you know what the microscope does to the data. And then there's two algorithms in there. There's a Richardson-Lucy, which is the sort of very standard one, and there's also an iterative constraint Tikhonov model. They're pretty much standard algorithms that you. There's also a Wiener filtering in there, but Both the ICTM and the Richardson-Lucy have a positivity constraint, and that's about it, other than the PSF knowledge. Yep.
Do you know of other biology or biohistoric groups using this library, and do you have time to evangelize, basically?
I don't have time to evangelize, really, and there's a few groups who use this, but are basically people who I've had collaborations with in the past. And they're mostly using it for localization microscopy, but they're using it a little bit for confocal data as well. It's still a little bit raw. I've sort of developed it to meet my needs and dumped it on Google Code, and so it's I think it almost is, but at this stage I think it's ready for distribution to people who know a little bit about Python and are prepared to sort of wade through some of the dependencies to get it installed. But I haven't been doing much evangelism.
It's just really frustrating to see lots of biology groups who use proprietary software that's sometimes very poor and that has huge claims.
Yeah, I mean, that's pretty much, basically, I see people using things like a mirror and stuff like that. And that's why I tend to write everything I want to do. It finds its way into here eventually. So I'm more a user, but I'm a user that doesn't trust a mirror or doesn't trust all these sort of proprietary nominal image processing software, which... Yep. I do. There's a partial list up in... do I have a list of microscope hardware which is supported? And the answer is that I do, but it's partial. But there's a partial list up on the Google Code site. The short answer is that it's and/or cameras, some sensor cam cameras, most PI piezos and translation stages, and there's a few other bits in there as well. If there's enough demand, I'd like to add the support for the Micromanager C API. Yep.
Would you consider a GPL or a VST licensed version or a partial version?
Yeah, I'm kind of constrained because I use things like FFTW, which are GPL, and there's a few other GPL things in there. But I would absolutely be prepared to partially license different things. I think at this stage I'd like to do that on a request basis. If you sort of see something you'd like, I'm more than happy to make a BSD. I probably won't make the whole package BSD, because at the moment there is also a commercial aspect in that software, at the moment people sell sort of instruments doing this for about 800K, and the difference between the instrument and the actual hardware that you need is about 700K. Or 600K, and that's basically in their commercial software. And so I don't want a company to just take this and run with it.
One more question.